Hi, Dr. Wang here. Today I'm going to do a short lecture on what is restriction enzyme. This is an enzyme that we as molecular biologists used all the time in the lab to create artificial DNA molecules. However, this enzyme is not something invented by scientists. It's something that already exists in nature. Its function is to defend the organism against the invasion of foreign DNA molecules. So for example, we have an E. coli cell over here, and it's being infected by a virus. So the virus is going to inject its viral DNA. I'm going to represent that with a blue line into the E. coli cell and try to replicate the virus. So here, suppose this is the viral DNA. It try to replicate itself within E. coli, and E. coli, of course, want to prevent this from happening. So what E. coli does is to use a set of enzymes called restriction enzymes to cut those viral DNA into pieces. So you can think of restriction enzyme as scissors. So how does this enzyme actually cut DNA into pieces? So for that, we need to look at the DNA structure first. This is a structure that we're already familiar with, right? So here, the two highlighted structure are the two strands of the DNA running anti-parallel to each other, and they are held together by base pairing in between. So we can see that the two neighboring nucleotides, one is here, the other is here, are held together by phosphodiester bond. So restriction enzyme is going to come in and break this phosphodiester bond open. So this is going to open up the strand. So restriction enzyme is considered as endonuclease. So if we have a DNA strand over here, restriction enzyme can cut this DNA right in the middle. So this is called endo. The opposite to this is called exonuclease. We have seen exonuclease when we discussed replication mechanism. So exonuclease also dissolve phosphodiester bond, but it can only chew up the DNA from the ends. So it basically works on exposed end, and it's going to chew one nucleotide off one at a time from the end of the DNA molecule, while endonuclease, such as restriction enzyme, can dissolve this connection right in the middle of the DNA. That's why it's called endo. So restriction enzyme recognize specific DNA sequences before it cuts the DNA. This is not a random cut. And that specific sequence is called recognition site. Sometimes it's also called the restriction site. So here we have an example of the recognition site from an enzyme called BAMH1. So what's interesting about this sequence is that this is a palindrome sequence. So what is a palindrome? Palindrome is when you read it this way and then that way, the sequences are identical. So here, we can fill up the sequence at the bottom, right? So here we have from 5 prime. Oh, wait, sorry. I think I have a mistake here. So let me relabel the direction. So the top strand runs from 5 to 3. The bottom strand should run from 5 to 3 in this direction. So here, opposite to C, we should have a G, right? So, and then G, and then A, T, C, C. So when you read the sequence from the top from 5 to 3, it's G, G, A, T, C, C. When you do this to the bottom strand, it's G, G, A, T, C, C as well. So this is called a palindrome sequence. And for BAMH1, it's going to cut the DNA strand open between the two Gs. So it's going to cut here, and it's also going to cut here. The result of it is that both strands are broken open, right? So and then the result is basically this, and I can actually pull this away a little bit. So this is the result of BAMH1 digestion. All right, so the next question we're trying to address is how often does a restriction enzyme cut DNA? So we can start with something that's a lot simple. So we can start with a palindrome site that looks like this. So how about just AT? AT is a palindrome sequence as well, because if you read the bottom strand, is also AT, right? So what are the chances of a sequence AT occurring just by random chance in a given sequence? The probability of having A at the first location is 1 out of 4, right? There are only 4 bases, so having A occurring by random chance is 1 out of 4. And having T occurring by random chance at the second position is also 1 out of 4. And then having AT sequence appearing by random chance is the first probability multiply the second probability. So this is 1 out of 14. So this is the probability of having sequence AT occurring just by random chance. So if I gave you a sequence that's 1,000 base pair long, for example, and then asking you, so how many times is this enzyme that cuts AT going to cut this sequence? Then to calculate this, you're going to simply take 1,000 and then multiply that by this. And then you're going to get a number that's how many cuts, right? So this is what are the probability of something occurring by random chance? It's 1 out of 16. So if the total sequence is 1,000, then it's simply 1,000 multiply 1 out of 14. So let me actually calculate, calculate this. All right, so if I get my calculator ready, 1,000 divided by 16. So this is 62.5. So I'm going to say that's roughly 62 cuts. So we can see that this is going to cut very frequently just because the recognition site is so short that this site occurring just by random chance is going to be high, therefore it's going to cut a lot more times. So what if we have something that cuts, for example, 1 out of 4 to the power of 4, right? So this is AT. So if we have an enzyme that's AATT, then this probability is basically this multiply 4 times. So then we can continue the calculation, and then we're going to see that this is going to give us close to 4 times, right? So here, 4 cuts. So here we can see that this enzyme with a shorter recognition site is going to cut more often. We often call this a frequent cutter. And then this enzyme that cuts a 4 base pair sequence is going to cut less time. So this is a less frequent cutter. So this is how we calculate how often does a restriction enzyme cut the DNA. And you can actually take this up. One more level is what about the average size then? So if we have, right, so a 1,000 base pair, and then we know that it's going to get cut 4 times. And then this is going to give you, so 2 base pair, right? So the average size resulting from this digestion is going to be 250 base pair. All right, so here the next concept that we're going to go through is sticky ends versus blunt ends. So as we can see here, after the BAMH1 cut, this is the resulting ends. So this ends looks interesting because we have a portion of the DNA sequence that's single-stranded. Right? So it's basically hanging out there. So this is called an overhang. So how do we distinguish 5' overhang from 3' from blunt? So I'm going to make an example here. So for the I have 3 sequences here. All of those are GGA-TCC. So the first enzyme cuts right here. All right. So here I'm going to use my eraser to erase one of the fragments right here. Right. So here. So after the cut, I get 2 ends. Right. So I can erase one of the 2. So this is what's left behind. And now I'm going to label the ends, 5 prime, and then this is 3 prime. So we can see that this single-stranded structure that's hanging over there is 5 prime. So this is called the 5 prime sticky end. And oftentimes we call this a 5 prime overhang. All right, so now I'm going to do a similar exercise, but now I'm going to cut between the 2 C's. All right, so instead of cutting between 2 G, as the blue example, I'm going to cut in between the 2 C's. So similarly, I'm going to use an eraser to remove one of the 2 resulting ends. So the next thing I'm going to do is to label the ends. So this is going to be 3 prime and this is going to be 5 prime. So we can see that this single-stranded DNA structure is now at the 3 prime end. So this is called the 3 prime sticky. So what is blunt end? Blunt end looks like this, cuts in between A and P. So when I erase one of the 2 ends, you can see that there is no single-stranded structure hanging out. So this end is blunt. All right, so here, this is called blunt end. All right, so here, the sticky ends are interesting because sticky ends will allow us to take the fragments of DNA and put them back together. So this is called compatible ends, right? So over here, I'm going to do this cut right here. So so here we have 2 recognition sites. The first one is GGA-TCC, cuts in between the 2 Gs. The second one is called CGAT, CG, also palindrome, right? Cut between CG and CG. So here, I'm going to take my eraser and then remove one of the 2 ends, right? So over here, I'm going to remove this part. All right, so here we can see that now we have two sticky ends. So here, we can actually put them together. All right, so here, if I take this green ends and then move it across like so, you can see that they fit perfectly together, right? So, and then this is because, right, so G paired with C, A paired with T, G pairs with C. So here you can actually take a ligase that ligate this 2, and then you're done. So this is basically cut and paste. So restriction enzyme will allow scientists to cut DNA at specific sites, generating fragments, while ligase will allow us to put those pieces back together and therefore creating artificial DNA. So how do we tell whether 2 ends are compatible to each other, right? So from the first glance, they're very different. But if you look closely, you will see that they do have something in common, and that's the 4 bases right in the middle are identical. Because those 4 bases participate in base pairing. This is how the 4 bases are put back together. So for 2 restriction enzymes to be compatible with each other, their recognition sites have to be identical right in the middle. Those 4 base pairs in the middle should be identical. They're both GATC, so they can be put back together. And another similarity is that they both cut in between the first and the second nucleotide. Right, so then it's going to generate very similar ends, and then that can be put back together. So here, down here are basically illustrations of how restriction enzyme works. We can see that here is a plasmid DNA, and we often use those as vectors to contain gene of interest. So the first step is to cut it open with restriction enzyme, right? So an enzyme that cuts between G and A is going to generate a 5' sticky end. So we can also cut the gene of interest in this case, represented by the pink fragment with the same enzyme. So that generates compatible end. We can see this AATT and this TTAA can come together and form base pairing. So once that occurs, we have a structure that looks like that. And the next time the ligase can come in and then seal that little opening. So basically making continuous DNA strands. So the result of that is inserting a piece of gene of interest into a vector DNA. And this is how the plasmid DNA that we analyze in the lab were generated in the first place, right? So this is the empty vector. It's only 4,900 base pair long. And then into it, we inserted something that's close to 5,000 base pair long. This is the coding sequence for MSH2. So then the resulting vector is 9,000 base pair long, right? So, and this is done through restriction digestion and ligation.